All right. Well, it looks like the Zoom room is filling up and the auditorium here is filling up, and it's great to see everybody. Thanks so very, very much coming, either in person or on Zoom. It's just great to see you all. And we have a very special Dean's lecture today from Priya Duggal, and really looking forward to her presentation. And I will acknowledge that her family is here, her parents, her husband, her sons. So welcome to you, and thanks for coming. But before I officially introduce our esteemed speaker today, I'd just like to remind everybody, why we're here. Several years ago, we established the Dean's Lectures to allow us to hear from our colleagues who have been recently promoted or appointed to the title of professor or senior scientist. And it's an opportunity for us to engage and get to know what our colleagues are doing, because often we don't get that chance. And I find it particularly enjoyable to hear about all the great work that our professors and senior scientists are doing. So I think everybody looks forward to these Dean's Lectures. And today I'm delighted to introduce Dr. Priya Duggal. Priya is a professor in the Department of Epidemiology with a joint appointment in international health. She is specifically focused in her work on genetic epidemiology and is one of the few genetic epidemiologists who cross fields with the genetic components of both infectious diseases as well as non-communicable diseases. She earned her MPH and her PhD right here at the Bloomberg School, and worked at NIH before joining our faculty in 2008. She's now the vice chair for faculty in her department of epidemiology. She also led the genetic epidemiology track for a number of years and remains a mentor, an important mentor for many of the non-tenured faculty in that program. Priya's colleagues will tell you that she is someone they can depend on. Someone who always steps up and always follows through. She is admired for being a generous and creative collaborator who always brings a fresh perspective to every project that she works on. Now, there aren't a lot of people like Priya who can speak equally well about the nuances of advanced genetic epidemiologic methods as well as the mechanisms of infectious diseases, and that makes her a hot commodity, if you will, for people who want to do work in this area. Priya will tell you that she was drawn to the field of infectious disease because she saw very deep needs all around the world. People were suffering from illnesses and conditions for which there were no treatments. This was an area of public health where there's a lot of questions and yet preciously few answers, and Priya was anxious to look for those answers. In her time at the Bloomberg School, Priya has studied hepatitis B, hepatitis C, HIV, and the enterovirus, as well as enteric infections that affect young children who are living with poor sanitary conditions. She currently leads the genetics arm of the ECHO study, which is gathering incredible data on the interactions of the environment and genetics from some 40,000 children and their mothers. And as I'm sure you know, Priya is also co-leading the school's long COVID study. This is a vital survey that will help us better understand the long-term effects of COVID-19, which for many people around the world are proving to be lasting and in many cases very debilitating. The team is aiming to reach 25,000 individuals, and they are well on their way to getting there. So we're really looking forward to the results of that work. As you can tell, Priya's sort of busy. She's working on some exceptional projects that will provide meaningful data. And as Priya put it, those data can serve as a voice for people who are suffering. They can articulate the nature of the problems before us and very importantly, point us towards potential solutions. Now, of course, we know these solutions don't happen overnight. For Priya and her colleagues in particular, it may take 20 years of research to gather the information we need to actually make a difference, to find those solutions. That kind of work takes patience, determination, and vision. But it has the potential to make an extraordinary impact, and we are so grateful for Priya's commitment to uncovering the knowledge that can touch lives around the world. Just to give one more example, there were lots of great examples of Priya's work. She has been involved with an effort to better understand acute flaccid myelitis, a condition that causes sudden paralysis, which begun surging in children around 2014. Working with her colleague, Dr. Erin Millstone, Priya has sought funding, launched a genetic study, and looked for links to polio by talking to polio survivors. No one asked them to do this work. They sought it out because they saw young people who desperately needed help. Their efforts mean everything to those parents and their families. As one father of a young boy put it, he said, "Priya is one of the only people investigating this problem. I'm just so grateful that someone was interested in our kids. It was like a miracle." Priya, we're so grateful for all you do for the Bloomberg School, but more importantly, for all you're doing for the public's health. So we're delighted to have you part of the Hopkins family. And so I will hand it over to Professor Duggal.
Thank you. Thank you. Thank you, Ellen. That was a very kind and humbling introduction. It's sort of amazing to think of all of the people that I've worked with for so long, and it is my privilege to speak to you today about some of my work. So, as Ellen said, there's a lot of different projects, and I'm going to highlight one of them in particular, sort of a larger one, to give you some insight into our work. But before I start, I saw this quote. So for anyone who has read The Plague by Albert Camus, I'm pretty sure I had to read it in French when I was in college as well. I just wanted to start with this quote. So everybody knows that pestilences have a way of recurring in the world, yet somehow we find it hard to believe in ones that crash down on our heads from a blue sky. There have been as many plagues as war in history, yet always plagues and war take people equally by surprise. And I think as we are living through a pandemic and have had years that have been exceptionally difficult for many people, it's important to remember, and I'm going to hopefully highlight this, that just like war, plagues have left their mark on us. And we're going to talk a little bit about that mark that has been left on us over the years. And so I co-teach a course to the doctoral students in the second year. And when we teach that course, we often talk about asking faculty to come in and talk about sort of their origin story of how they've come into the field of epidemiology. We also ask them to talk about the questions that how do you ask a question? How do you think about a question? So I thought I'd start there. I came to Johns Hopkins to get my master's in public health, in international health. And my advisor was Richard Morrow, and I had a meeting with him in the first couple of weeks of school. And at that point, he sort of sat me down and said, and for anyone who knew Richard Morrow, an unbelievably sweet man, exceptionally kind and very smart. And he said, I think it would be really smart if you got a degree in epidemiology or biostatistics, or maybe even went to medical school. That times have changed for us a bit in terms of how we did international health before. And as you can see in your MPH class, you have health ministers that are coming to Hopkins to get a degree. And so it might be advisable for you to get some tools, and then you go back out into the world. And so that is the advice I took. It was brilliant in those first couple of weeks, perhaps a bit unsettling because this is where I had come to do this work. But at the same time, I was right across the hallway in East Wing Auditorium, for those of you who remember East Wing before it was Sheldon Hall. And I had the privilege of having Leon Gordis teach us epidemiology, the principles of epidemiology. And anyone who heard Leon speak knows that he had an unbelievable ability to draw you into epidemiology, really through humor, often quite canned humor, but also to make it really accessible, to make epidemiology easy. And I didn't know too much about epidemiology. I knew there were hospital epidemiologists, but I think that sitting in that classroom and hearing what Dr. Morrow said and sort of being overwhelmed with an amazing MPH class, epi made a lot of sense. Of course, for anyone who took that series, you took the second term with Dr. Sklo, and that's when he turned everything on its head, and all that easy stuff in epi became all this nuance that we deal with on a regular basis. But I came here with a background in genetics. And so I came to my colleagues in genetics, Terry Beatty and the recently deceased Gloria Peterson. And I said, I'd like to do something in this area of genetic epidemiology that I had sort of just learned about. And this is right at the time as we were transitioning in genetics from sort of Mendelian-focused single monogenic disorders into large population scale disorders. And we could make that change because technology was changing. It was allowing us an opportunity to look at larger scales of data. And so the recommendation, and I know Terry's on this call, was that I should focus on one of these types of outcomes, right? I should look at things like heart disease, which is a major killer of people in the United States, or cancer, like some of my other colleagues focused on. And I was a little stubborn. I came here to do international health. So this wasn't necessarily where I wanted my focus to be. And when we think about public health overall globally, you can see that these green bars are infectious diseases or neonatal conditions, often infectious oriented in life. And so outside of a lot of countries, you still have people dying, because these are mortality statistics from 2019, go back to 2000, right? In the late 90s, you have people dying from infectious diseases. So my goal really was sort of to merge those two concepts. And so with the tutelage of Terry Beatty and David Vlahov, who ran the Alive Clinic and the Alive Study, I was able to do my dissertation in this realm. And that's really set off all of the work that I have done. And so with that, I'm going to start with my thank yous. So I mentioned some people already. That's Terry Beatty, my PhD advisor, Joan Bailey Wilson, my postdoc advisor, both emeritus as of today, and both amazing trailblazers, both for women and geneticists in the field. Just amazing as a woman to have two women who trained me unbelievably. Then Dave Thomas, Bill Petrie, Rashidul Haq, and Selma Geronimo, who I've been working with for 20 years, basically. And it is really quite amazing, the mentorship they've given me on the research side and the openness that they've come to with genetics overall. And I've added in there David Celentano, although we've never worked together. I was John Sammet's last hire in this department, which means I was David Celentano's first faculty member. And in that role, he has given me a tremendous amount of opportunities to grow outside of research. And I thank you for those opportunities. In the center row right here, you're going to see a whole bunch of people. Those are genetic epidemiologists. They're amazing people. They're my friends. They're the people that have been with us, some who I trained with, others who have been here, and of course, two who are now deceased, Linda Cow and Gloria Peterson. And then right here on the bottom, with the exception of Genevieve Wojcik right in the center, are all the doctoral students that I've trained, and they are amazing people. And I am lucky today that I'm going to present some of the work by Genevieve, Dylan, and Rebecca. So thank you. Everything you hear is a result of these people, and they are amazing. So, and behind them are lots of other people that I'm not even talking about. Okay. So when we think about infectious diseases, what do we think about? Well, our focus really globally is on disrupting transmission and treating people either at risk of disease or infection so that we can limit morbidity, mortality, and subsequent spread. That's our goal. That's what we do in our daily basis. That's what we want to get at. And in order to do this, we have to understand infection. And the best way to understand infection for an infectious disease epidemiologist is to understand the natural history of disease. When we look at the natural history of disease, we know that not everyone who gets exposed to something gets the outcome or gets disease. And it's really that question that I want to understand and figure out. So if we think about things like TB and hepatitis B, both on the ends of this study, we know that about a third of the world's population has been exposed to them, but only about 10% of people go on to a chronic state that's symptomatic. And if you think about something like measles, if measles had been in this room 2 hours before we had all joined this room and none of us were vaccinated, 85% of us would walk out with a measles infection. But it's really that 25 to 30% of us would have symptomatic complications from that measles. So what makes those individuals different than the people who didn't even get measles and were sitting in the same room as us, and the people who had sort of milder symptoms? And of course, we all understand COVID, right? At the start of COVID pre-vaccine, about 15% of people ended up in the hospital. And the question is, who ends up in the hospital? And so we can ask some questions, and they focus often about looking at the host, and we can think about normal things like age and sex, having a comorbidity, having a co-infection that drive you either closer to a more severe outcome or faster to progression. And you can think about that with COVID, right? We knew that the people getting hospitalized early on were on the population scale, men who were older, who had a litany of comorbidities. But any clinician in this room that saw a patient during that time knows that those ICUs were also filled with women who were younger and who didn't have those comorbidities. So what explains those individuals being there? And others who may have gotten COVID, who were older and male and had a comorbidity, never had to go into the hospital. So how do we explain those differences? And often people will say, well, it's about the pathogen then. That's what it's about. It's about how much you got in terms of your dose, the variance you got, and the exposure. And I think that's true. It's often really important for us to think about the pathogen itself and what happens. But are there examples where we can sort of learn about that pathogen and dose? And unfortunately, they're usually tragedies. So in 1926, in Lübeck, Germany, 251 babies were injected with the same live dose of virulent tuberculosis instead of BCG. Clearly a tragedy and an accident. The babies were too young to have significant prior exposure to mycobacteria, and they were not previously vaccinated to BCG. They all got the same strain. They all got the same dose. 72 babies died, and 173 babies survived. So what made those kids who survived different than the kids who ended up dead? And so that is sort of the question that we ask. And then one a little bit closer to home, because of all the polio work that's been done here at Hopkins, is the Cutter incident, where activated polio vaccine was accidentally administered instead of inactivated because of poor quality conditions where the vaccine was being made to school-aged children. 200,000 children were injected. 40,000 ended up with infectious symptoms, 200 with paralytic polio, and 10 of those kids died. But what about those 160,000 children that didn't even have any symptoms, right? So that's the question overall that we're trying to ask. And so for me as a genetic epidemiologist, I'm trying to figure out how do we determine if it's playing a role? And one way we can do it is sort of ruling out, as I said, the role of a pathogen dose and environment not playing a major role. Another way is to think about the disease heterogeneity, and we often think, as I just described to you, about sex and age and comorbidities, which are biologic conditions. But we can't ignore some of the social conditions about poverty or racism or access to care, right? That plays into that heterogeneity that we sometimes see with outcomes. So we need to account for that, or at least acknowledge that that may be driving it. And then the last is, for any genetic epidemiologist, we try to assess familial aggregation. That's us saying, does this cluster within a family? So you can sort of imagine someone looking at breast cancer, for example, and you talk to a clinician and you say, okay, yes, my mother had breast cancer, all my maternal aunts had breast cancer, my grandmother on my mom's side had breast cancer. And so we call that a clustering within that family. And there's elevated aggregation, and we could calculate what we call a heritability, or what's attributable to the genetics. But that's a little tougher to do if we're looking at an infectious disease. Let's pretend now it's influenza that I'm looking at, and someone comes in and says, well, I had it, and my kids had it, and my husband had it. That clustering has very little to do with genetics, or so we think, and a lot more to do with the fact that you're talking about a contagious disease. So how do we assess familial aggregation? And one way we can do this is in twin studies, and this has successfully been done. I'm going to show you an example, but the one I really wanted to show you was one that George Comstock did here at Hopkins on TB. It's a beautiful study looking at trying to understand the heritability of TB using twins. But we'll do this one as well. So this is a study looking at poor response to HPV vaccine, that's the surface antibody response, at 1 year of age in sets of twins. There were 370. And 20% of the twins seemed to show a poor response. And so they looked at the correlation of the antibody response between monozygotic, that's identical twins, and dizygotic twins, that's siblings. So from a genetic perspective, monozygotic or identical share the same DNA. So we expect them to have the same outcome or phenotype. Dizygotic twins are the same as siblings. So on average, you're sharing about 50% of your DNA. So we expect you to have some similarity, but not as much as a monozygotic. So when the monozygotic sharing is greater than the dizygotic sharing, it suggests that a genetic component is at play, and we can calculate that heritability. In this case, the monozygotic sharing was 0.65, and the dizygotic is 0.35, and we can calculate a heritability, or the amount attributable to genetics, at about 60%. right? So something as simple as your response to a vaccine can be calculated and we can evaluate it. So that's one way we can look at aggregation. So given that, the way we would do things next is we would say to ourselves, let's conduct a genetic study. We've shown aggregation, we've accounted for heterogeneity, or we think we have. How do we do that study? So there are 4 letters that we care about the most in genetic epidemiology, and that's A, C, T, and G. They make up your strands of DNA. And between all of us, we have, each of us and every one of our cells has 6 billion of those letters that are there. And between me and any one of you, except my family, we share 99.6% of our DNA. It's a little higher with your family members, right? And so that's about 4 to 5 million differences that we're looking for overall. And so we can actually sequence you as an individual, or we could do an array where we do it every so many kb, and we're capturing a good portion of the genome. And then we compare the people who are cases and the people who are controls. You can also do this in a family-based design. So it's actually pretty simple, right? The hard part is trying to identify who's a case and who's control, trying to figure out the study initially from an epidemiologic perspective, and then genotyping them and doing all those pieces. But the basic analysis for a simple genetic epi study is like a chi-square, except that you're doing it 1,000,000 times, right? Or a logistic regression, you're just doing it lots and lots of times. And so that's how we do it. We do it for cancer or heart disease, for oral clefts, whatever you're doing, that's what we do. But for infectious diseases, where we have a little bit of a problem is defining our controls and our cases. And that's because of exposure. Because I need to know that you were exposed to a disease before I can say that you are a control, right? I need to know that you had a chance to have something and your body said, nope, I'm not a case, right? If I just take someone who was locked up during COVID and never saw anyone, then they're not really my control, because they never had a chance to be exposed to something that's happening. So to me, and to all the epidemiologists in the room, this should seem like a really simple concept. It's like our basic idea of cases and controls, and we say, do they represent the population that they're coming from? Are they similar? But somehow this concept has gotten lost in the world of genetics, and that's because we've had this huge advent of biobanks, and we've had this huge advent of electronic medical records. And so that has been amazing for people who study cancer and heart disease and diabetes, because from those records, you can get information about tests that people have had and who they are. But we're not screening people for infectious diseases, especially ones that can occur very frequently, right? How many times are you going into the physician to get specific tests? You're not. And so if we rely on those types of databases for our data, we're sort of falsely assessing people and saying that they're a control when they're not a control. And so because of that, it led to some work that a doctoral student, Dylan Dushan, led, because we sort of had to answer it. We kept saying it over and over again, and no one really listened to us. So we said, let's actually show it. Let's show it with some real data. And we also showed it with a simulation. So just to orient you, we're going to just follow this panel here on the left, right here, I think. You can see it. And this is 20,000 cases and 20,000 population-based controls. And the yellow blocks are actually representing well-characterized controls, coming from a cohort study, coming from some place that we know is real. And the red blocks are actually representing people who are population-based controls. I could be a mix. You could have been exposed. You may not have been exposed. I have no idea. I just know that you're considered a control. And so at the very top, when the prevalence of disease is 100% in the population, think something like Epstein-Barr virus over a certain age. Then it doesn't matter if you use a control that is well characterized or use a population-based control. But as the population prevalence decreases over time and you go to 75%, 50%, 25%, and 5%, you can see that that red box shifts over, right? And what that means by it shifting is that that odds ratio is increasing when it shouldn't be changing. And it's telling us that there's a spurious association that's occurring. And with that spurious association, I now believe that there are new genetic variants out there that support something that doesn't exist, right? And so we have to pay attention. The same is true if we took what we think are more normal biobank sets of 20 versus 200,000 population controls, where we see the exact same effect with just greater significance of what we're looking at. So because controls matter, it means we're going to have to spend a lot of time looking at studies and making sure that we've captured people's information really, really well. So I'm going to tell you a story about that, specifically as it relates to diarrheal or enteric infections. So in 2000, 1.25 million children died annually from diarrhea. And in 2019, we're fortunate that that number really was halved to 500,000 children who died annually from diarrhea. And this is just a map sort of showing you the places that are most affected by diarrhea globally. And you can see that Africa and Asia are really suffering overall with children who are dying. So why is there so much diarrhea-associated mortality, and how did we get that number down? We got that first, what are the risk factors? So poor sanitation, water sources, lack of hand washing, poverty, undernutrition or malnutrition, and then a series of pathogens. And these pathogens are the ones that we know have the highest attributable risk for diarrheal-associated mortality overall. So how did we get it down? The first thing that people did was actually change food security in countries. We were able to get people better nourished by getting them more food. And we're talking about children under 5, right? So that was one major measure. There's also been huge WASH measures, including dealing with sanitation and hand washing, that have been in place for the last 20 years, where we're trying to get that number down. But we're still dealing with 500,000 kids who are dying, right? It's a lot of kids. And so along with all of this that's happening, we're also seeing this other cycle, which is this sort of vicious cycle of kids who were born malnourished or undernourished, who then are much more susceptible to an infectious disease or an infectious pathogen, and then they go on that cycle and they get more malnourished. Or children who are born more nourished, they have tons of assaults on their enteric system, they get more malnourished, and we go right back into that cycle. So it's about disrupting this particular cycle. Now, this figure comes from the Malnutrition and Enteric Disease Study, also known as Malady, of which Hopkins Department of International Health played a huge role. It's 8 global sites that were specifically looking at malnutrition and enteric disease. And that's because undernutrition affects 20% of children in the developing world, but it attributes to 50% of the deaths in children less than 5 years. And of course, enteropathogens, not only are they causing diarrhea, but they're also causing intestinal inflammation, changes to that intestinal barrier, and absorption issues, which makes it more difficult for you to get the calories that you need overall for a child to be healthy. But the part that I think everyone in Malady was most struck by is that these kids end up stunted. So by the time they're 5 years old, they're exceptionally stunted in their growth. And on top of that, by the time they're 2, you can already see cognitive deficits, meaning the things that we could do to help these children, we no longer can do. They're cognitively deficient by the age of 2 years. Overall. So there's got to be something we can do to fix it. And that's sort of why we can think about genetics, because we're going to think a little bit outside the box, the traditional box that we've thought about. We can't literally pick up kids and move them to another country, although people have suggested that. But we can maybe think about other roles that are at play. And so this work takes place in Mirpur, Dhaka, Bangladesh. So the first plot shows you where Bangladesh is, and the second one is focusing here on Mirpur, which is outlined in red. I don't know if you can see this when I move it, but this sort of red outline. So Mirpur is actually a population of about 1 million people. It's considered a slum population. And over the last 20 years, actually, you've seen a change where you actually have middle-class individuals also in this population. But you can see these images here, it's still got a lot of slum population within it. Right? So this is a very large population of individuals overall, and that's where all of our studies are going to be focused. So my colleagues at the University of Virginia, Bill Petrie, and at the International Center for Diarrheal Disease Research in Bangladesh, an amazing center with huge ties to Hopkins, led in this study by Rashid Al-Haq, have set up several birth and child cohorts. Now I'm really lucky. So Bill came to Hopkins pretty much, I think, a couple days after I turned my dissertation in before I defended. And he came to speak to Terry Beatty about looking at familial aggregation and talking about people at Hopkins because they were the experts. And Terry said, Priya, don't you do infectious diseases? I think you should come into this meeting. And since then, we've been working together on these studies. So I've been really fortunate to be a part of the building of most of these studies. And so the parents and the children are visited every 2 to 3 days by healthcare workers for details about any diarrheal episodes the child may have. If diarrhea is detected, the child is examined, a stool sample is collected, we test for enteropathogens, surveillance stool samples are collected every month, and blood samples are collected from the children every 4 months to test for serum antibodies. It's a lot of work for those healthcare workers to visit these families so frequently. And we're fortunate that there are a couple more studies led by Poonam Korpi that are now emerging with new cohorts. But those don't yet have genetic data. So I'm going to focus on these 3 that do. The first is the DACA birth cohort. It was actually an arm of the malady study that I mentioned before, one of the global sites. It was designed to study the malnutrition on child development. It looks at 626 infants enrolled within the first week after birth, and then followed forward up to 5 years. The second is a clinical trial designed to look at rotavirus and oral polio vaccine uptake in DACA, and it has 700 children and their mothers followed for 2 years of life. And again, 2 to 3 times a week. And the third one is the Cryptosporidia birth cohort, which is an observational cohort, again, with sites in Mirpur. That's one site, and then one that's a little bit more rural in Mirzapur. So working with these 3 sites, we're going to ask some questions about the role genetics plays with diarrheal disease. And so what are some of the things we're looking at? Well, these are some of the enteropathogens of interest, and these are the ones that have some of the more higher attributable risk to morbidity or mortality. So since I think most of you don't think about diarrhea on a regular basis, and I advise that you don't, it's important that you understand exactly what is going on. So we've got bacteria like Campylobacter and Shigella that can lead to bloody diarrhea, fever, prolonged duration, and really high frequency during that of 6 episodes within 24 hours. Now remember, we're talking not about us, we're talking about infants. We're talking about children in the first year of life having these types of assaults. And so we can treat these with antibiotics, but we also know that a growing public health issue is antimicrobial resistance. And so these antibiotics may or may not work in these populations. The next is viruses, and I've picked 2 here to highlight. One is rotavirus. Anyone with a young child is aware of it. You've had them vaccinated. Anyone with an older child remembers the days when you didn't have a child vaccinated. It's very easy for a child to catch rotavirus in the United States prior to vaccine, and very easy in these populations. It is the #1 contribution to death. So that vaccine was huge. Astrovirus is a virus that doesn't seem to cause that many problems overall. But when you look at statistics of what is circulating in kids in their stool, diarrheal stool, astrovirus is present all the time. It's sort of like Giardia. It just sort of hangs out there in really high numbers, and it can be symptomatic. And then we have 2 protozoa, Cryptosporidium and Entamoeba histolytica, and they can lead to extra intestinal issues like amoebic liver abscess, and they can also be quite severe. And there are treatments available, but these treatments aren't necessarily recommended for children under the age of 1. And so we don't actually have a treatment that's available. And so actually Dr. Poonam Korpi is going to lead a study looking at a clinical trial trying to address some of this work. So this is what we're looking at. This is what we're seeing. And I'm going to tell you a story about one, and that's Entamoeba histolytica, and then expand on the others. So what do we know about Entamoeba histolytica? Well, we know the transmission is fecal-oral. We know that once you ingest it, the protozoa goes through a trophocyte stage, and then it can get encystized, and it can actually be excreted as a cyst and start that whole process over again. But we also know that it can lead to things like I mentioned, amoebic liver abscess, specifically in adult males, and it can live with people for a really long time. These 2 images are sort of showing you the clinical presentation of the colon when you have it. And what's sort of fascinating, and the reason that I put it up there, is you can sort of see how it's ulcerated. These pictures could also be pictures of inflammatory bowel disease, because actually they are easily misconstrued for each other. In the literature overall, unless you know someone's history of travel, for example, you wouldn't necessarily be able to distinguish them overall and when you're looking at it. And so for our study, and I need all the genetic epidemiologists to just take a breath when I show you my numbers, but we're dealing with 170 cases of Entamoeba histolytica total and 574 with no histolytica, for a total of 744 people. Let me just say that if someone came to me right now and said they wanted to do a study like this, I would tell them they were crazy. There's no power in that study. I don't know why you're thinking that. But I hope that I've set this story up enough to show you the desperate times call for desperate measures. It makes us think outside the box about what might be available and what we could do. And what are we going to lose, right? If we can get funding for it, and we did, what would be the problem in seeing what happened? Well, in this case, no problems whatsoever. So for those of you not familiar with this plot, it's called a Manhattan plot, and it's meant to mimic the skyscrapers in Manhattan. Now, this is not a view of Manhattan, but it is a beautiful view of a city with one big tower in it. So pick your favorite city that has a tower in it, and that's what you're going to see. Each of these on the bottom axis, on the x-axis, are your chromosomes going from 1 to 22, and each little dot represents a test for association with a single nucleotide polymorphism, those A, C, T, and G differences that we were talking about before. And then what we've plotted is the minus log p-value of that test of association, whether it be a logistic regression or a chi-square or whatever we're looking at. So the reason we're looking for skyscrapers is the more significant it is, the taller those buildings are going to be. Right? And then because we're doing over 1 million tests, we have an issue of multiple testing. And so we have that dashed line there that's representing our threshold of significance, accounting for the multiple tests that we've actually done. And so lo and behold, we have a peak, and we have a genome-wide peak. And then we looked at our cohort separately in this forest plot and the meta-analysis. And of course, now all of those genetic epidemiologists should be so sad when they see that my odds ratios are in the range of 2.4 or 2.5. These are huge, huge effects. These are massive effects overall that you're looking at, right? When you think about stuff for heart disease, we're looking at 1.05, right? We're looking at a 2.5-fold increased risk of having entamoeba if you carry this particular variant. So what do we know about this variant? So this is just a zoomed in plot here on the left of that same thing that you looked at. And now we've just put at the top the genes. So we're looking at CUL2 and CREM and CCNY. And then right below it is data taken from the GTEx portals. That's looking at expression data for every single one of those variants in that region. And the gold represents CREM. And that one black line you see there represents CUL2. So it looks like the genes expressed in this area are all being linked up to CREM. And so what do we know about CREM? We know it's a cyclic AMP responsive element modulator that regulates genes via cyclic AMP response elements, or CREs, in the promoters. That's a lot, especially for an epi audience. But we hypothesized that CREM is activated by, in this case, entamoeba histolytica-induced cyclic AMP signal transduction. Okay, so somehow, using this gene, entamoeba is activating a signal transduction pathway. And our colleagues at the University of Virginia set out to prove that, and we're fortunate that they did. So they see that actually E. histolytica does activate cyclic AMP signal transduction. They see first that it peaks in 2.5 hours in intestinal epithelial cells, plateaus at about 4.5 hours. That's that straight line that you see right there. And then we looked at cells silenced for CREM. So we did an siRNA on them, which silences the cells all together. And those black boxes you're looking at are the CREM that's been silenced. And you can see that there's overall in the E. histolytica group a decrease in what you're looking at. So there's some interaction between E. histolytica and CREM. And then CREM is also expressed early in infection in mice, and that's happening within the first 12 hours. We're seeing that expression that's occurring. Okay, so then they went forward and they actually did some knockout mice, and we knocked out the CREM in those mice, and we saw that the CREM, that's the one on the CREM knockouts, that's the KO that's listed there, actually had more infection than the wild type. And then they did the same thing to look at apoptosis, and they see that the knockouts have more apoptosis than the wild type. So a lot of evidence here to suggest that there's a role for CREM, and this is just the histology showing you the wild type and then the knockout with the apoptotic death of the cell. Okay, so we were excited about this. This was really awesome. This is telling us that there's something at play here, and we were able to find something with 170 cases that could translate to something overall that has an impact for children that don't really have an option on treatment. But it didn't sort of stop there, because then when we did a little bit more investigating, we said, well, what do we know about the loci that are specifically this CREM locus? If we take this CREM locus, what do we know is out there from other studies? Because the infectious world is pretty small compared to everyone else who's done work. And so this is a big table, but I want to say the SNPs come first. That's that variant we're looking at. And then the functional class, and then the reference, the disease, the risk alleles, the p-values, the odds ratios, and then our work over here in comparison. And what kept coming up is that this exact gene region is associated with inflammatory bowel disease, Crohn's disease, and ulcerative colitis. So I told you they look really similar in terms of their clinical presentation. They also are controlled. IBD has a lot of genes that control it, but one of the genes that controls it is the same as what's controlling Entamoeba histolytica. So there seems to be some pleiotropy, or the control of by one region on two different phenotypes, which is going to be part of our story. So we did this. That's right there. Now this slide to me is a tough one because this is going to represent more than 15 years of work in one slide, because in the interest of time, I can't tell you all the stories that I would love to tell you. So I told you one to make you familiar with what we're doing. And so that's Entamoeba histolytica, an odds ratio of 2.5, some clear findings in the lab of what we think is supportive, and some interesting appreciation of what's going on with autoimmunity. And then Genevieve Wojcik in her postdoc years led this on cryptosporidium, and we see another peak, a totally different peak, but another peak. This is protein kinase C alpha, and both of these resulted in a great amount of funding for our collaborators to follow these up in the lab, because these are diseases that need treatment, and these are diseases that we can target. And again, an odds ratio of 2.5, and so we should be thinking, of course I'm finding the big stuff, because that's all I'm powered to find, right? But there is big stuff out there. That's the point. And then Shigella. So Shigella is a nasty little one that we're looking at, and it can actually lead to high amounts of mortality. And we see this is work that Dylan Dushin did as a side project. We tell our doctoral students to do side projects. This was an awesome side project. So we found two variants here, and I'm going to focus on the one on the right, KRTP18. It's actually a pseudogene. So when we first saw this, we were like, I wonder what that means. No idea. And then we dug a little bit deeper, and you'll see that this is a protective effect. You're 2.5 times less likely to get Shigella if you carry that keratin 18 pseudogene. So when I think of keratin, I think about hair products, but apparently that's not a good mix. Keratin actually has a huge role to play in our immune system, and also in our structural system and our cellular system. And in this case, keratin 18, which has 85% similarity to the pseudogene, is involved in the Shigella injectosome, or the Shigella secretory system. Just happens to be that it is critical for Shigella to enter the cell. And so this may be explaining why we see this association, and it may have indicated that we found something else that's important for Shigella entry. And this is Campylobacter. So this is work led by Rebecca Monday as part of her dissertation work. And she found this region here on chromosome 8 with ARGF10 that also has associations. It's also protective against Campylobacter. And then this work was led by a master's student, Laura Chen, who is now about to graduate from medical school at the University of Maryland. Also sad that we never published this particular piece, and that's because we couldn't make sense of anything that was going on with this. And so Rebecca took it on to sort of reinvestigate, working with Laura, and they've identified these two genes that are important. And now, several years later, we can see they have clear chromatin interactions that are involved with the immune system, and specifically with the gut. And so that's also put at risk. So a lot of work and a lot of separate GWASs, but a couple of take-home points. You don't see me repeating any genes here. Each and every single one of this is in the same sets of those 3 populations of kids, some variants of them, and yet the same gene isn't popping up for all these diarrheal pathogens. It's a different set of genes for each one. And so why is that? And how is that possible that I have genome-wide significant hits for all of them, right? That's sort of unheard of when we think about things. But we're not asking the question that we started with, and that's this question on diarrhea. That's where we started. We didn't start with the pathogens. We wanted to ask, what is going on with diarrhea? So we said, let's do a GWAS on diarrhea, but we expect to see what we saw in the pathogens, right? That's what we expect to see. We expect to see the most prevalent pathogens will be the ones that come up in the GWAS hits, and we'll just sort of see a recapitulation of that data. So Rebecca has led this, and I feel bad. I'm stealing this before she presents it for her dissertation work, but she looked at diarrhea in 2 different ways. First, diarrheal episodes in the first year of life, just counts of them, and then days of diarrhea. And that's important because we count a diarrheal episode. You have to clear that infection, and then we count it again. And she took extremes. So she took people who've never had a case of diarrhea, those kids. Anyone who's a parent, think about your kids in the first year of life never having a diarrheal infection. That's hard to imagine in the United States. These kids live in abject poverty, and those kids don't have a reported case of diarrhea in the first year of life. And then the second group, 6+ diarrheal episodes in the first year of life, which is a huge assault on their system, or 25 days or more of diarrhea overall. Okay, what do we find? Well, the one on the left is no diarrhea versus 6+ doses, and the one on the right is looking at no diarrhea versus 25 days of days of diarrhea. And so we see 3 genes that emerged on the left. The AP00959 is actually a non-coding RNA that has really interesting chromatin interactions with NCAM2, which is a neuronal adhesion molecule that's involved in the development of the enteric nervous system, and it's downregulated by ulcerative colitis. It's also been seen in other models with Hirschsprung's disease because of Hirschsprung's relationship with NCAM2. And then SAMD12 is highly expressed in the gut. And then over here, we also see this non-coding RNA pop up. So there seems to be some similarity between the two, but another gene that popped up called WSCD1 that is downregulated in pediatric inflammatory bowel disease. Okay, so that's a lot. And again, I keep going back to this idea of what we see. The one recurring theme is sort of inflammatory bowel disease keeps making a play. So in sum, for this part, every pathogen seems to have their own genome-wide significant locus. The pathogens don't match up with extreme diarrhea, suggesting that we're looking at a different mechanism. Small sample sizes mean big results if you characterize your population correctly, meaning you have good controls and you have good cases, meaning you have a good cohort. And genetics is playing an important role in the immune system and infectious diseases. But I'm led to ask the question of how did we find these things? It's not really making a lot of sense in the world of genetic epi, except that we have this concept of selective pressure. And that's the thing we have to remember with an infectious disease, that the largest selective pressure on the human genome, so we're thinking now Darwin, right, is pathogens. I might say that maybe climate change is also going to have a huge impact indirectly, right? But overall, pathogens have had the strongest pressure on the human genome. And so we can think of some examples. I can think of cholera. And the Ganges Delta region has the highest level of cholera in the world. And that has a lot to do with the number of floods that occur in that region. And yet, when you look at that region, they have really low levels of blood type O. And so that's fascinating because blood type O is a highly susceptible for severe cholera thing that you have. So if you have blood type O, you are highly susceptible to severe cholera. And yet in this region, we have exceptionally low levels of blood type O. And that's because all of us are the products of our ancestors. And that means that we are the products of the people who survived what came before us. And we're here to tell the story in our genes, even if we don't know the story that came before us. And we see that with malaria and balancing selection with sickle cell disease so that people who have sickled cells are more likely to survive through malaria. But of course, that sets them up for sickle cell disease. And we see selective pressure. And I think this one's an interesting one because of the ethical issues associated with it. And that's HIV and West Nile. So HIV, in order for it to enter the cell, needs CD4. And it also needs either chemokine receptor 5, CCR5, or CXCR4. For R5 tropic virus to enter, it was noted in the mid-90s that people who lacked 32 bases in the CCR5 receptor had a non-functional CCR5. And because it was non-functional, HIV couldn't enter the cell. And so despite repeated exposures, those individuals didn't seem to get HIV. And that was pretty awesome. Those folks called them their blood gold, right? Because they could do anything they wanted and they weren't going to get HIV. And then you had people that ended up getting it through another entry point, and that's X4 tropic virus. And the cohorts here at Hopkins contributed to all of that original findings. But, so that's great. That has led to a drug being developed that targeted CCR5. That led to a non-ethical man in China, a scientist who decided to target twins, babies that were being born, and delete their CCR5 because he said, oh, God forbid, maybe in the future they could get HIV. This way they won't get it. right? He did that, and everyone was in an uproar about the ethics of it. And the idea that he kept saying is, well, it's benign. I can get rid of your CCR5 and you'll be protected from disease. But lo and behold, we have West Nile virus, a flavivirus that's emerged in the last 80 or so years, that people who have the Delta 32 base pair deletion, you're 8 to 13 times more likely to have severe West Nile virus and die from it than individuals. So you're protected from HIV, but you're at high risk for West Nile virus. So no change that we're making in the human genome is necessarily going to be one that's benign, right? There's going to be a trade-off. And that's this idea here that these resistant mutations that we look at may protect us against some infectious diseases, but they also come with a cost. So HLA-B57, great for HIV control. HLA-B57 leads to psoriasis. FUT2, great to protect you against norovirus, seems to lead to Crohn's disease. CFH protects you against meningitis. It was the first gene identified for the first GWAS that was done for macular degeneration, so you might go blind. APOL1, exceptionally dear to the hearts of our faculty here because this is sleeping sickness, predominantly seen in Africa and individuals of African ancestry. The late Linda Cow found its association with end-stage renal disease. So those individuals are protected from sleeping sickness, but high risk for end-stage renal disease. And then ERAP2, this is a really beautiful study that just came out from the University of Chicago at the end of last year, where they looked at samples that were fossilized pre the Black Death. And they see that the allele frequency changes over time. And so you were protected from Black Death, but now you're at risk for Crohn's disease. So there isn't sort of, it's great. Protection is awesome and we want that so we can survive, but it's likely coming at a risk. And that risk seems to be more driven in the chronic disease world and autoimmunity. And so we took our specific traits and we looked at what we call a PheWAS. That means you take your trait and you look to see, is it associated with any other phenotype that's out there? And interestingly, for cryptosporidiosis and for astrovirus, it's associated with metabolic traits. And there's been long discussions about the role of metabolic traits and obesity with infectious diseases and many different theories about its play. But I think this opens up a different discussion about, can something protect you or put you at risk that's outside of just traditionally being obese or being malnourished? And then, of course, I mentioned the one of Craminea histolytica and its association with autoimmune diseases and the GI tract. Okay, so because of that, I'm going to finish up and tell you our future directions. We're hoping to look at pleiotropy of enteric loci with autoimmunity and metabolic traits. We're hoping to extend beyond Bangladesh. So we've been parts of networks for years that are in different parts of the world, and they likely have had different selective pressures that have been placed on them. And so I think it's important for us to do our due diligence and look at those regions where they also have high levels of diarrhea. But it's also important to think about that diarrheal finding that I showed you that's protective against kids having diarrhea, and think about looking at it in a study like ECHO. We don't have the extremes in the United States like we do in these populations, but what if we see that prevalence that's high? Because wouldn't it be great if we could target that for treatments for diarrhea that were outside of just oral rehydration solutions? And then we're also looking at the evaluation of the gut microbiome and metabolomics with genomics and sort of that interaction and what is at play between them. And then the interaction of host and pathogen genomes has always been what we've wanted to do, but it takes a lot more work to get the sequence of the virus or the pathogen intermixed with that of the host. So I'm going to end in two ways. One, so that was the end of my story, but as you heard, we also do some other work. And so I want to start with this one real quick. And this is Albert Camus again. Many continued, hoping that the epidemic would soon die out and they and their families be spared. Thus they felt under no obligation to make any change in their habits as yet. Plague was an unwelcome visitant, bound to take its leave one day as unexpectedly as it had come. I mean, we've lived through this pandemic and we know that so much of this is true, right? That people have not necessarily been willing to make changes in their life. And I think if you've survived and you've made it this far, that's great. But we also have this thing called long COVID. And so in 2020, not that far after we were dealing with the initial craziness of 2020, Brian Lau, Shruti Metha, and I were discussing patients, discussing anecdotal evidence, and we were really witnessing that participants were telling us from our varied studies that they had continued long-term symptoms. And so we waited and we talked and we discussed, and at some point we decided that we were the grown-ups and that we couldn't sit at the kids' table any longer, and we couldn't wait for someone else to do it. And so we launched the Long COVID study, which is a virtual cohort aimed at getting 25,000 people. We have 21,000 consented to date, 12,000 have agreed to be contacted for further follow-up, and 6,000 have completed their first round of that follow-up. And so I just want to present just a couple of points to you because I think it's really important. And that's that when you ask patients, and that's what this plot is at the top, the orange is long COVID, the green is resolved COVID. So you had it and you're doing great. You ask people, are you 100% of what you were before? 80% of people who are resolved say yes, right? And then you can sort of see that it goes down a little bit in the next categories. But only like 5% of people who have long COVID tell you that they were 100% of what they were before. And actually, you even have 5% who are telling you they're 1 to 10% of what they were before they had their COVID infection. So it's just having a huge effect on people. And then this is just data where we took the extremes. We took individuals who have what we classified as disability by disability scales. And there are 4 different sort of categories here. One is walking a quarter mile. That's like walking here to the Washington Street garage. One is walking 10 steps. That's up 1 flight to the 2nd floor. Doing heavy housework like vacuuming and light housework like dusting. And what you'll see that we have as comparison, no COVID at the top, resolved COVID and long COVID. Because the truth is we're living through a pandemic. And so there's a lot of things that can affect you and harm you in terms of your mental capacity as well as your physical capacity in this time. But you can see the orange means you're unable to do it. The yellow means lots of difficulty. And that's really only highly prevalent in that last category of long COVID for each of those categories. And this is looking at what we call the Woods Mental Fatigue Inventory. So that's looking at your mental fatigue and cognitive function. And most people are going to fall on the scale, if we took it in this room and you didn't have a COVID issue, somewhere between 5 and 10. And if you look at these box plots, the yellow is long COVID, you're seeing that they far exceed it, right? Doesn't mean that people who didn't have COVID are also having mental fatigue issues or having other issues. They are. You can see those examples. But on average, these people with long COVID are having a huge effect. So if you haven't registered for our study, please do. That's the website. This is what we feel like we do all the time to tell you to participate. We need people who've had COVID, people who haven't had COVID. We need people who've resolved COVID. If you're 18 years or older, you can enroll. And the reason it matters is because this is public health. This is what we do. And realistically, that's the only way we can get everything done. So thank you so much. I'm delighted that my family could be here today. Those are my 2 boys and my husband and my parents who are in the audience and my little dog who came during COVID that is precious to all of us. So thank you very much. And I'm happy to answer any questions.
Thank you. That was a phenomenal talk, and you have many stories to tell, and they're just fascinating. And thank you for all this work. It's just a huge body of work that is continuing. It's great, fantastic. So we'll open up the floor for questions, and if anybody on the Zoom would like to ask a question, should they email you, Becky, or
they can check, send me a message in the chat.
send Becky Newcomer a message in the chat, and we'll call on you, and you can ask your question. Or if you want to just raise your yellow hand, that would work too. But yes, Elizabeth.
I'd love to hear an update on your reflexive myelitis research. I remember at the beginning, we had this like surge in cases, and it was really scary. And you were starting to do work on like the genetics of it at the time.
Yeah.
Now it's like the number of cases has fallen.
Yep.
I'm curious to hear what you learned in your research and whether there's still a problem.
Yeah, so
Great.
thanks, Elizabeth. Thank you. Thank you. So Elizabeth's talking about acute flaccid myelitis. So it's basically like polio in our current day. So children are exposed, we believe, to enterovirus D68. And Andy's in the room, so he can back me up on this. We believe it's enterovirus D68. And after they are exposed to it, a certain percentage, the numbers are pretty much like polio, 1 in 100,000, end up paralyzed. It was pretty shocking in 2014. We saw cases not really here in Maryland, but nationally. And then the case numbers started to increase in 2016 and 2018. And looking at that trend, we were all anticipating that 2020 was going to be just this crazy year. And of course, COVID hit before the season had started for enterovirus to circulate. And so we didn't see it. And so it was delayed because people were, at home, and delayed into 2021. Kids weren't in school, because this is affecting children. And then 2022, we still didn't see it. And now the discussion sort of is, why aren't we seeing it? That's sort of this idea, because we do know that enterovirus D68 is circulating in large numbers, and yet we're not really seeing those cases. And so there's some discussion about it being the type of enterovirus D68, its role, and how it has adapted or evolved to be different. But we had launched our genetic study. A lot of it was put on hold because of our need to, we'd had to launch a COVID study at the same time on COVID gene. And so we put a lot of it on hold, but we are hoping to get right back into it to sort of analyze it. We've sequenced the kids that we've collected, and we have over 100 families that are enrolled in our study.
Other questions? Yeah.
Those genes that you're identifying in diarrheal diseases, I should be able in some of them to do network analysis to see what is the cascade of genetic events and eventually pathogens.
Yeah, so no, Fidel, we haven't done a really great network analysis across it. I think that we're at the point where now we can sort of take all of those genes, especially as we start to see the roles that they're playing, and sort of ask those questions. On an individual scale, we've looked at those genes, but not sort of on the grander scale of what could be happening in the gut overall. But I think it's a great idea.
Yeah.
Thank you for a great talk. There are also different factors involved here, which is the genetics of the infectious agent. I'm sure that you know a lot of women who are infected with human papillomavirus, unless D6B7 is expressed, they don't get cancer. So it's what's involved in the host that, unlike BRCA1, BRCA2 activates the viral gene, which leads to cancer.
Yeah, I think that's such an important point, and one that I didn't highlight enough, which is that I focused here today on the human genome side of them, but the pathogen genome, maybe not as large, but it is just as complicated in terms of its interaction with the host. And so some of our work they're doing with hepatitis B, for example, is looking at that interaction between hepatitis B and the human genome. And that's not easy. I will tell you, it is not easy to sort of make heads or tails because of the depth of which you're going to have to sequence hepatitis B, and how you're going to align some of those hepatitis B reference genomes, right? So when we think about viral genetics, for example, we use very few references to align everything to, but there's actually a lot going on. So work that I did with Poonam with cryptosporidiosis in Bangladesh just showed us there's a lot of variation of even cryptosporidia in our populations, right? Not all of it necessarily functionally different, but a lot of variation that has gone underappreciated because we haven't been sequencing it. So I think the ability to take a MySeq or something into the field and sequence is going to change things dramatically in terms of what's actually circulating at that time and its specific interaction with the host and really immunity of the host. Yes.
So what do you see as some of the steps down the line? Is it to use this to screen people in order to treat? Is it in terms of preventing the infection, the preventable attributable risk seems to be more in the prevent exposure versus the interaction with the genetics.
Yes.
What do you see as the downward?
Yes. So I think that ideally, right, that this will be out of our hands in the sense that we hope that people will take the findings and the genes that we identify for the development of treatment, of vaccine or therapeutics. So really pushing this out to say, look, we're looking at things that have huge effects, because that's a 2.5 times odds ratio for one allele. If you carry both alleles, that's a 5-fold difference. It's huge. And so if we can find these important landmarks, these important genes that are involved, perhaps that will spur some drug development or therapeutic treatment development for diseases that really don't have great options overall. They've got some options, but they're not great options. And that's true really for all infectious diseases. We make this argument with hepatitis B that we've got a vaccine, but we don't necessarily have cure for a lot of people who are suffering. So can we do something that's adjuvant or will improve the life of individuals? But I don't see it as being used to screen. I don't think it's feasible to go into a developing country and say, let's screen your population and see who's at risk. I think it's got to be that you take this and then you develop.
Right, yeah.
Yeah, so I'm complete genetics dummy and I learned a lot from your talk.
Good.
So you had an example of how the mutations of the protective against infectious disease seem to have negative effect in terms of autoimmune disease. Was it just selected examples or it's a general pattern you see?
So I don't think so I selected those examples, but those examples are the ones that are out there in the sense that, like, I don't know of other examples where you see protection from an infectious disease and protection from an autoimmune disease. When we see protection from an immune disease, we seem to see susceptibility to the autoimmune disease. And I think that that's that interplay that we've been fascinated with for a long time of immunity and autoimmunity, right? How does it play out? And I think the part that this study sort of brings to the forefront is that we haven't been looking at things like enteric infections when we think about IBD, inflammatory bowel disease. People just haven't thought about it. They haven't thought about these sort of chronic infections that are occurring in kids for a long period of time and what effect it then has on the human genome, but also you take people out of that environment or you get rid of those pathogens. How does the body react to that over time? I don't mean, overnight, but over time, how does it react?
Yeah, one more question. Yeah.
Fantastic talk.
Thanks, London.
You mentioned at the beginning of your talk about that, the issue with using the bowel bank and studying big bowel bank controls to study, the genetic association for infectious disease. As you know, like in COVID-19, they have gone ahead and done the big bowel bank studies, and now they have found 10, 15 or different loci. Could you tell us, are they biased, somehow because of they did not, take the exposed control, or how they have affected those discoveries?
Yeah, so actually, Nanjan, that's what drove us having to write the paper, right? Because we, Genevieve Wojcik and I had lots of conversations with those groups that were doing it, and it just seemed to be lost overall in the general community. I don't mean in those individuals, that exposure was really, really important. And so, yeah, I think that there are biases in those results. I also think that the big ones, chromosome 3 and others, are real because we've seen the data where they've restricted it. In those reviews, we made sure that they restricted it to people where you had a clear exposure and yet you didn't have it and you still see it. So I don't say that all of those results are biased. I think some of them are real, but I think now they've got like 40 genes that have been identified and some of them are not going to be real. And I don't think we can, I think in the time of pandemic, a lot of things are excused, but now that we can go back, we sort of have to say to ourselves, what's real and what's not real, because otherwise lab scientists are going to go in and look at those genes and they're going to say, this is an area that we should all be focused on. And they're going to spend a lot of money following down a path that maybe isn't that real. So maybe just a reevaluation of that type of data.
Great. Well, again, Priya, thanks so very much for an outstanding lecture and for everything you're doing. I would like to invite everybody, at least in this room, sorry for the folks online, but we do have a reception out by the Wall of Wonder, and please join us and we can continue the conversation there. Thanks again for coming.
Thank you. Thank you.